Adaptations to the environment in C4 and CAM plants. The key points you should understand by the end of this presentation are as follows. The process of photosynthesis in desert plants has evolved over time to help conserve water. Plants that use C4 photosynthesis separate the initial fixation of carbon dioxide and the Calvin cycle spatially. Plants that use crassulacean acid metabolism separate the initial fixation of carbon dioxide and the Calvin cycle temporally. Some key terms to keep in mind throughout the lesson are C3 photosynthesis, C4 photosynthesis, and crassulacean acid metabolism. This process is also known as CAM photosynthesis. To understand the adaptations of C3 photosynthesis and CAM photosynthesis, we must first understand the basics of C3 photosynthesis. Plants that undergo C3 photosynthesis are called C3 plants, and these are considered normal plants, meaning they have no photosynthetic adaptations to the environment. C3 photosynthesis only happens in the mesophyll cells of the plant. In the C3 pathway, we have CO2 entering into the mesophyll cell. Here we have the fixation of carbon dioxide by rubisco. This produces 2 3-carbon molecules of 3-phosphoglyceric acid, or 3-PGA for short. Here inside the Calvin cycle, we begin to produce sugar. The excess 3-PGA is going to produce more rubisco biphosphate. This is then going to be recycled again to fix more carbon dioxide and sugar. This sugar is going to be in the form of CH2O. Again, it is important to note that this reaction only happens inside of the mesophyll cells of the plant. Some examples of plants that undergo C3 photosynthesis include wheat, rice, millet, peanuts, cotton, and most trees. In fact, about 85% of the plants in the world are C3 plants. Plants that undergo C4 photosynthesis are found in different environments than plants that undergo C3 photosynthesis. Because of this, they've evolved a different type of pathway. Here, the light-dependent reactions in the Calvin cycle are separated spatially. This means that the light-dependent reactions occur inside the mesophyll cells, while the Calvin cycle occurs inside the bundle sheath cells. The bundle sheath cells are the special cells found around the leaf vein. Again, in the CO4 pathway, there are 2 types of reactions are found in the different cells. We have the mesophyll cell, and we also have the bundle sheath cell. In the first step of this pathway, CO2 from the stomata is fixed to a 3-carbon compound called phosphenol pyruvate to produce a 4-carbon compound called oxaloacetate. An enzyme called PEP carboxylase catalyzes this reaction. PEP carboxylase fixes carbon very efficiently, so the C4 plants do not have to keep their stomata open as much. In this next step, The oxaloacetate is converted to another 4-carbon compound called malate. The conversion to malate requires the reduction by NADPH to NADP+. Next, the malate exits the mesophyll cell and enters the chloroplast of the bundle sheath cell. One of the products of the decarboxylated malate is CO2. This CO2 combines with ribulose biphosphate and goes through the Calvin cycle to produce sugar. The other two byproducts of the decarboxylated malate are pyruvate and NADPH. The pyruvate re-enters the mesophyll cell, reacts with the ATP, and is converted back to phosphenolpyruvate. C4 plants are found in hot and sunny environments, so they've had to create quite a lot of adaptations to overcome this. One of those adaptations is the mesophyll cells constantly are pumping carbon dioxide into the bundle sheath cells in the form of malate. This means that there's always a higher amount of carbon dioxide to O2 around rubisco. This is a really important strategy to help minimize photorespiration. The benefits of reduced respiration exceed the cost of the ATP required to move CO2 from the mesophyll to the bundle sheath cell. About 3% of all vascular plants are C4 plants. Some of these include corn, sugarcane, crabgrass, Bermuda grass, and many other grasses, and sorghum. CAM photosynthesis is also known as crassulacean acid metabolism. These reactions are separated temporally, meaning that the initial CO2 fixation occurs during the nighttime cycle, and the malic acid to Calvin cycle reactions occur during the daytime. At night, CAM plants open their stomata, allowing CO2 to diffuse into the leaves. CO2 is fixed into oxaloacetate by pep carboxylase. This is the same reaction that occurs in C4 plants. The oxaloacetate is then converted to malate. In CAM plants, it isn't always converted into malate. Sometimes it's other organic acids. This malate or organic acid is then stored in the vacuole until the next morning. In the daylight, CAM plants do not open their stomata, but still continue photosynthesis. The organic acids are transported out of the vacuole and broken down, which releases carbon dioxide. The carbon dioxide enters the Calvin cycle. There's a high concentration of carbon dioxide that is maintained around rubisco. The Calvin cycle produces sugars, and the remaining byproducts of the malate are recycled to produce phosphenolpyruvate. CAM plants are dominant to very hot and dry areas such as deserts, so they must adapt to avoid photorespiration but also be very water efficient. Humidity tends to be higher at night and temperatures tend to be cooler. That's why opening the stomata at nighttime helps factor in to reduce water loss and avoid photorespiration. Some examples of cam plants are pineapple, agave, orchids, cacti, kalanchoe, and the family Crassulaceae, which was actually one of the first families that this cam photosynthesis was discovered in. Overall, all three pathways use the Calvin cycle to generate sugar from carbon dioxide. Each pathway is best adapted for a different environment. In C3 plants, CO2 fixation and the Calvin cycle are not separated, and the stomata are open during the day. C3 plants are best adapted to cool and wet environments. In C4 plants, the initial carbon dioxide fixation and Calvin cycle are separated spatially, and the stomata are open during the day. C4 plants are best adapted to hot and sunny environments. In cam plants, the initial carbon dioxide fixation and Calvin cycle are separated temporally. The stomata are open during the night in cam plants. Cam plants are best adapted to very hot and dry environments. Because of the hot environment that C4 and cam plants are both found in, both plants have adapted over 25 times to overcome the loss of water by photorespiration.